Hello students, welcome back after the break. So we were discussing about the tertiary structure of protein and the supersecondary motifs which form the basis for structural classification of proteins. Before I went for a break, I left a very important activity for you all. I asked you all to make a search or to think about the various ways in which the elements of secondary structure can be arranged to form supersecondary motifs. Now let us find an answer to that. So what are supersecondary motifs? As we have studied, that is for the first part of my lecture I have already discussed that the combination of the elements of secondary structure are first placed into different groups or classes called supersecondary motifs. Supersecondary motifs include the elements of secondary structure and the adjacent connections between them. Now next to this, the elements of secondary structure and the adjacent supersecondary motifs are further compacted and folded to form a globular lobe-like structure called the domain. A peptide, every peptide chain of a polypeptide consists of multiple domains, and the spatial relationship between all these domains present in a single peptide chain of a polypeptide makes up what is called or takes up a structure what is called the 3-degree or the tertiary structure of a protein. Hence, for a monomeric protein which consists of a single peptide chain, the tertiary structure of peptide is the tertiary structure of the entire protein. But for an oligomeric and multimeric protein containing many peptide chains, every peptide chain will have its own 3-degree or tertiary structure. Now let us return back to the way the elements of secondary structure are arranged to form supersecondary motifs. Now supersecondary motifs include, that is actually they include or are the elements of secondary structure, the adjacent elements of secondary structure, and the neighboring connections between them. The supersecondary motifs form the basis for structural classification of proteins, SCOP, and therein lies a database called SCOP database, which borrows a scheme that is well known, meant for classifying the structures of proteins. According to SCOP database, all protein structures of all proteins or all protein structures are divided into 4 different classes depending upon the way the elements of secondary structure are connected or joined together to form supersecondary motifs of those proteins. So depending upon the way the elements of depending upon the constituent elements of secondary structure and in accordance with the SCOP database, all protein structures are divided into 4 different classes. That is, the 4 classes are all alpha, which are in which the supersecondary motifs are only made up of alpha helices, all beta, in which the supersecondary motifs are only made up of parallel and anti-parallel forms of beta pleated sheets, alpha beta, in which the supersecondary motifs are made up of alpha and beta segments that are interspersed or intervened, and alpha plus beta, in which the supersecondary motifs have regions of both alpha helix and beta pleated sheet that is well connected together. So according to that, this is the SCOP data where the classification of different protein structures into 4 groups according to SCOP database. Now let us take a look over different supersecondary motifs or different classes of supersecondary motifs which form the basis for this classification of protein structures into 4 different classes. There are a variety of supersecondary motifs. Basically, there are 4 different classes of supersecondary motifs which are a combination of the elements of secondary structure. The first one is the beta alpha beta motif. As the name suggests, it's made up of both alpha helix and beta pleated sheet. It is the most popular, is the most frequent supersecondary motif that is found in globular proteins. It consists of a right-handed alpha helix joining to parallel strands of beta pleated sheet, such that both the strands of beta pleated sheet have similar N-terminal or amino terminal to C, that is carboxyl terminal, channel orientation. Now next to this comes another supersecondary motif that is called beta meander motif or beta hairpin motif. What is beta meander motif? It is the second most frequent supersecondary motif next to beta alpha beta motif that is found in globular proteins. Beta meander motif consists of antiparallel strands of beta pleated sheet which are linked together by hydrogen bonds. Now it assumes a structure which is the same as that of, or which looks more like a hairpin, hence it's also called beta hairpin motif. Now what is the third one? Now the third one is the supersecondary motif that is found in fibrous proteins. It is alpha alpha motif. Alpha alpha motif is found in alpha keratin and is found in various other fibrous proteins, but is not found in globular or the regulatory proteins like hemoglobin, myoglobin, lysozyme, cytochrome C, etc. So what is the structural arrangement of alpha alpha supersecondary motif? It consists of two alpha helices which are joined together by a loop such that the alpha helices are antiparallel to each other and are properly oriented with respect to one another. Now next, the fourth class of the supersecondary motif consists of beta barrel motif. So beta barrel motif is frequently found in proteins like human serum albumin, in nucleotide binding proteins like glyceraldehyde 3-phosphate dehydrogenase, etc. So what is the structural arrangement of the supersecondary motif? The beta barrel motif is actually made up of parallel or antiparallel strands of beta pleated sheets which roll up upon each other, or which are rolled up together to form a cylindrical barrel shaped form. So that is this is what is called a beta barrel motif. Now again, there are various subtypes and subforms of beta barrel motif according to SCOP database. Then beta barrel motif, the most common forms of beta barrel motifs are beta meander barrel motif, that is beta meander barrel, then Greek key motif, then alpha beta barrel motif, etc. Beta meander barrel, and as the name suggests, it has a structure quite similar to that of beta hairpin motif or beta meander motif. Beta meander barrel has its polypeptide backbone arranged in the form of beta meander made up of antiparallel strands of beta pleated sheet. Now Greek key motif is the next, is another, or is one more very popular beta barrel motif. In Greek key motif has in its backbone antiparallel beta pleated sheets which are joined together by large loops such that it assumes a structure which is similar to that of ancient day Greek key pottery, hence the name Greek key motif. Because it bears a structural resemblance with Greek key, ancient day Greek key pottery, it has been the barrel or the motif has been named as, that is here has been named so, or has been named as Greek key motif. The third one is the alpha beta barrel motif. Now it's a form of beta barrel motif. What is alpha beta barrel motif? It has a backbone arranged in the form of beta alpha motif, in which alpha helices run between parallel strands of beta pleated sheets. So these were the these were the three different subtypes of beta barrel motif. So to summarize, we have four different types of supersecondary motifs. That is beta alpha beta motif, which is the most frequent supersecondary motif in globular proteins. Next to that is beta meander motif or beta hairpin motif, which is the second most frequent, that is supersecondary motif found in globular proteins. Now third one is the alpha alpha motif, which is found not in globular but in fibrous proteins like alpha keratin, and is made up of alpha helices exclusively. And fourth one is beta barrel motif, which is a combination of the parallel and anti-parallel strands of beta pleated sheet rolled up to form a cylinder barrel shaped form, and has various subtypes like Greek key motif, Greek key beta barrel, then alpha beta barrel, then beta meander barrel, and so on. So now in order to there is one more database called, other than SCOP database, called CATH database. It uses more automated methods, more modern technology, and will provide you more information about the structural classification of proteins. It is CATH, or class, architecture, topology, and homologous superfamily database. So the database is providing information about supersecondary motifs, and hence the different classes of supersecondary motifs which we have discussed today, and about the four different groups into which the protein structures are, classes into which the protein structures are grouped, are the SCOP database, the structural classification of protein database, and the CATH database, that is class, architecture, topology, and homologous superfamily database. So hope you have gained enough information, or you hope the fundamental concepts of the supersecondary motifs and the structural classification of proteins are very clear to you. You might be having further queries, which of course I'll be addressing in the next session. And as a part of your activity for today, I'm leaving a question for you all. Just peep over the issue that what are the common database. Just try to find an answer to this question, that other than SCOP and CATH, what are other databases which do take a look over structural classification of proteins and are concerned with the supersecondary motifs of proteins. So see you all in the next class or in the next lecture. Stay tuned till then. Keep healthy, stay safe, keep studying. Goodbye.